Yes, so welcome to the class once again, students. So we are discussing mutations in the few classes that we are doing now, isn't it? अभी हम जो classes कर रहे हैं उसमें हम लोग mutations के बारे में पढ़ रहे हैं. In the previous class, that is part 1, we have discussed about the two chromosomal mutation, है ना. Mutation, what is mutation, ये हमने इसके पहले की क्लास में देख लिया है. Those who have missed, you can see the part 1 from there. If you want, I can add the link of that class, है ना, so that आपका continuity में रहेगा. So we have already discussed what is mutation. We have seen the types of mutation. हमने दो प्रकार के mutation देखे हैं actually as a flow chart, like chromosomal mutation or gene mutation. In chromosomal mutation, I told you that again you can get this chromosomal mutation can be depend upon the change in the number as well as structure, है ना. यदि नंबर चेंज हो रहा है तो हम उसको न्यूमेरिक म्यूटेशन बोलेंगे. यदि स्ट्रक्चर में चेंजेस हो रहे हैं हम उसको मॉर्फोलॉजिकल म्यूटेशन बोलेंगे, जिसमें yes, in this morphological or change in the structure, you can say deletion, duplication, inversion as well as translocation. Out of that deletion and duplication, ये दोनों चीज़ हमने इसके पहले की क्लास में बहुत ही अच्छे से पढ़ी है. We have discussed that along with examples, है ना. So the remaining two, that is inversion and translocation, that we will discuss in today's class. आज की क्लास में हम ये दोनों type of mutation or the chromosomal mutation we will discuss. Come on. So if I start with the third, because why I am saying third, क्योंकि deletion and duplication we have already completed in the previous class. Once again, the link of that class I will add in the description box. अच्छा, तो थर्ड वाला जो है जिसको हम बोलेंगे इन्वर्शन. इन्वर्शन हमको पता है व्हाट इज इन्वर्शन. इन्वर्ट होना, है ना, उल्टा हो जाना. सो एक्चुअली जब व्हेन द जीन इज रोटेटिंग एट 180 डिग्री, दैट मीन्स हम क्या बोलेंगे, वो इन्वर्ट हो गया. ऐसा है तो ऐसा हो गया, isn't it? The rotation for as a in a form of 180 degree, that is the what you call it as an inversion. So inversion involves the rotation of a part of chromosome or a set of genes by 180 degree on its own axis. Okay, now inversion जब भी होता है students, it involves the occurrence of breakage and reunion. यानी जब वो पार्ट ऑफ क्रोमोसोम रोटेट होगा तो कैसे रोटेट होगा, डेफिनेटली ब्रेक होगा, फिर रोटेट होगा और फिर वहां पे फिट हो जाएगा. सो इट इन्वॉल्व्स ब्रेकेज एंड रीयूनियन. गॉट इट स्टूडेंट्स? The net result of inversion is neither a gain or nor a loss in a genetic material. क्योंकि what is happening? The same set of genes are coming there, but yes, definitely they are inverted, so there is no loss. Deletion नहीं हो रहा है या फिर add भी नहीं हो रहा है extra का कुछ. No duplication. The genes are that only, but they are now rearranged, I can say. Isn't it? So there is not a gain or loss in the genetic material, but simply a rearrangement of a gene sequence. I hope this is clear. So we can say that the inversion किस तरह से हो सकता है. Suppose as I have given you here, you can see that the normal order of a segment with a chromosome 1, 2, 3, 4, 5, 6. For example, यदि हम बोल रहे हैं ये एक क्रोमोसोम है जिसमें नॉर्मल ऑर्डर क्या है 1, 2, 3, 4, 5, 6. ठीक है. Breaks occurs in the region 2, 3 and 5, 6. Come on, take it like this. Now I am making a rod like structure there. Suppose I am adding 1, 2, 3, 4, 5, 6. ये normal order है. Yeah. And the break occurs in region 2, 3 यहाँ पे and 5, 6 and 5, 6. सो क्या हो जाएगा, 2, 3 बोला और 5, 6 बोला है, ब्रेकेज यहाँ पर हो रहा है, यहाँ पे और यहाँ पे, isn't it? सो ये वाला जो पार्ट है यहाँ से कट हो जाएगा. दिस विल फर्स्ट इट विल बी ब्रेक, ओके, एंड देन इट विल रिवर्स. ठीक है, वहां से निकल गया, रोटेट हो गया 180 डिग्री, एंड देन इट विल अगेन गो एंड unite there. So what you will get after the inversion, the order आपका क्या चेंज हो जाएगा, 1 2 तो ऐसे ही रहेगा, 1 and 2 it will be as it is. Now here there is rotation, so what will come here, there will be 5, then it will be 4, and then it will be 3, and then here it is 6. So you can see the inverted chromosome, it will have the fragment or the segment as 1 2 5 4 3 6. तो यहाँ पे लॉस नहीं हो रहा है, यहाँ पर गेन नहीं हो रहा है, बट यहाँ पर क्या हो रहा है, इट इज रीअरेंजमेंट. I hope this is clear. Yeah, so this is in a simple way, this is what we call as an inversion. An inversion heterozygote has one chromosome in an inverted order and its homologue in a normal order. Now this is again an important thing. यदि हम heterozygote की बात कर रहे हैं, या और इन्वर्शन एक क्रोमोसोम में होगा, बिकॉज इफ आई एम टॉकिंग अबाउट अ हेटेरोजाइगोट, तो जो जिसमें इन्वर्शन होगा दैट विल बी इन दैट इट विल बी एन इन्वर्टेड ऑर्डर, लेकिन उसका जो होमोलॉग रहेगा, यस, वो कैसा मिलेगा, वो आपको नॉर्मल ऑर्डर में मिलेगा, 1 2 3 4 5 6. यस, देन यहाँ पर क्या हो जाएगा, क्रॉसिंग ओवर में इट इज पॉसिबल दैट देयर वुड बी सम सॉर्ट ऑफ प्रॉब्लम, और गड़बड़ हो जाएगी, isn't it? We will see how it happens one by one. I hope this much is clear. यहाँ तक समझ में आया? Yes, you have to keep this in your mind. Now, the location of inverted segment can be detected cytologically in the meiotic nuclei of such heterozygote. If we are talking about the heterozygote, तो inverted segment जो है वो आपको आप उसका लोकेशन फाइंड आउट कर सकते हैं. Yes, कैसे? By the presence of an inversion loop. As I told you कि यदि ये वाला सिचुएशन होता है, this is a situation and if I want to cross over, yeah, so crossing over में प्रॉब्लम हो जाएगा because the pair will not form. One is inverted, one is normal. तो इसमें जो इन्वर्टेड होगा ना, वो क्या करेगा, 3 को 3 के साथ लाना है, यदि मुझे 3 को यहाँ पर लाना है, 5 को यहाँ पर ले जाना है, तो व्हाट इट विल बी डन, यहाँ पर लूप फॉर्म होगा. यस, सपोज दिस इज माय, इन नॉर्मल केस दिस इज, इफ आई एम टॉकिंग अबाउट फ्रॉम हियर, यहाँ से मैं जा रही हूँ, सो दिस इज 3 एंड दिस इज 5. यस, नाउ दिस इज इन्वर्टेड, सो दिस इज 3 एंड दिस इज 5. Got it? If I want to make a crossover, so what will happen? This has to go here and this has to come here. So ये 3 3 आ गया, 5 5 आ गया. ये कब होगा? When there will be a formation of a loop. है ना? ये ऐसा है तो उसका क्या करेंगे, हम उसको लूप फॉर्म करेंगे, तो यहाँ वाला पार्ट इधर आ जाएगा और यहाँ वाला पार्ट यहाँ चला जाएगा. So there will be always a formation of a loop, student. That is an inversion loop. So when you will see this inversion loop, yes, in the meiotic nuclei, that means it is the location of inverted segment, there it is detected. I hope this is clear. Now this inversion यदि हो रहा है, yes, और ये loop formation जो हो रहा है, again यहाँ पर दो चीज, two things are important, whether this is with the centromere or without the centromere. The formation of inversion loop, yes, it includes the centromere or it do not include the centromere. On that basis, again, we can see that there are two types of inversion. Okay, so cytogenetics distinguish between the two types of inversion based on whether or not the inverted segment includes the chromosome centromere. Yeah, so there are two types of inversion on that basis. One is paracentric and another is pericentric inversion. ठीक है? So in short क्या पढ़ा हमने अभी तक आपको, what we have seen here, that yes, if I am talking about inversion, it is a product when there are two breaks. Inversion के लिए दो break होगी ना, जैसे हमने देखा. Here this is one break and this is another break. So 2 breaks will be there, yes, in a chromosome, and the intercalary segments, it will reunite. ये फिर से reunite हो गया यहाँ पर, but in a reverse order. Yes, but in a reverse order. So 2 breaks are necessary, and then 180-degree rotation is there in case of inversion. So 2 types of inversion if I say, paracentric and pericentric. अब क्या है ये paracentric and pericentric? Now inversion that do not include the centromere. जब inversion हो रहा है और उसमें centromere included नहीं है, तो हम उसको बोलेंगे paracentric. Okay, and if there is an inversion which includes the centromere, then it is called as a pericentric. So if it is centromere included, peri. Centromere not included, para. You have to remember this. आपको ये याद रखना है अच्छे से. Yeah, so when the centromere is not a part of the inversion, it is your paracentric inversion. Got it? Like if I am talking about A, B, C, yeah, you can see the inversion is here. यहाँ पे inversion हो रहा है. D, E, F में inversion है, तो यहाँ पर ये inversion यहीं पर हो गया है. This centromere, ये जो centromere है, ये involve नहीं है. तो जो भी हो रहा है वो centromere से अलग हटके हो रहा है, जो भी inversion हो रहा है. Yes, that is paracentric. In this case, this is the centromere. Yes, inversion is again there. Yes, you can see here, this is DC and this is B. Yeah, so AB जहाँ होना चाहिए CD, तो यदि यहाँ पर inversion हो रहा है BCD में, या ये normal BCD क्या हो जाना चाहिए DCB, तो DCB तो हो गया, it is inverted, but here the centromere is included. That means half inversion is on this side, half is on this side. Not half up, but yes, some part, the inversion you can see on the both sides. Centromere is included, that means. So this is called as a pericentric inversion. I hope this is clear. Very much clear or not? So what is paracentric and what is pericentric inversion? Again, I will add here, if I am talking about a paracentric inversion, that means remember centromere is not part of that. Yes, centromere is not included. जैसे कि मैंने अभी आपको एग्जाम्पल में ही बताया, if the inversion is there, A, B, C, D, E, F. Suppose inversion is here in this case. So that means after inversion what you will get? You will get A, B, C, F, E, D. Yes, so this is what is called as a paracentric. ये pericentric बोल रही हूँ मैं. If I am talking about the pericentric, again there what you will see, that means centromere included, break point within a gene. In this case, functions of a gene can be interrupted because breakpoint is there within it. Yes, so inversion of gene sequence around the centromere. Inversion around centromere. यहाँ पर कैसा होगा? I am making a very rough sketch, okay students, just to explain you. If I am talking about A, B, C, D, E, F, and if the inversion is between B, C, D, E, यहाँ पर इनके बीच में inversion है. Yeah, so what you can see here, क्या बोल सकते हो? इसका result आपको मिलेगा. It will be like A. Here it could be this D and C. Yes, D, C, and here it will be B, E, F. Yes. So this is what is called as a pericentric. I hope this is clear. One more important point I want to tell you, that all inversion, it is not necessary that all inversion causes damage. Yes, it is not necessary that all inversion causes damage, students. Again, I am repeating. Yeah, it is not necessary that all inversion causes damage. Now you will say, ma'am, how? Isn't it? कैसे? यदि inversion हो रहा है तो कुछ ना कुछ, because the cat, it is reverse there, है ना? So it will have a problem during the crossing over. Definitely the loop will form, but still. But students, why I am saying that all inversion will cause damage, it is not necessary. Like suppose, if this is the case, for example, if I am showing you two uh, thing there, yes, two, I can say A, B, C. Suppose this is A, B, C, two possibilities we will see, and this is D, E, F and G. Okay, I am making little bit इस तरह से, yes, so D, E, F and G. अब यहाँ पर inversion हुआ है. Yes, so during inversion what I told you, there should be the two cuts. Yes, if in this case the cut is, I will make a different color. Yeah, यदि cut is between the two genes, do not hinder any of them. यहाँ पर कट है लेकिन ये जीन्स के बीच में से नहीं है. सपोज ये कट यहाँ से हो रहा है और ये वाला कट यहाँ से हो रहा है. These are the genes. The white one, the white lines that I have drawn, that is the gene. But the cut is not between, it is not on E or it is not on D. It is not on F or it is not on G. It is in between. So this cut between the two genes, it is not hindering any of them. And if such genes, after such cut, they go inversion, yes, then it will show a normal response. क्यों? क्योंकि the gene is not disturbed. This F is as it is, this E is as it is. After inversion definitely आपको मिलेगा क्या? आपको मिलेगा yeah, after inversion you will get this A, B, C, D, F, E and G. This is inverted, but it is as it is not damaged. So this is normal. Yeah, because the locus is not damaged in this case. So this type of inversion, if it is there, then there is no problem. You will get a normal response. But, yeah, but this is the first possibility. But the second possibility, I will say that if the cut along during the inversion is across the gene or from the gene locus. Yes, same thing if I am drawing. This is A, B and C. This is D, E, F and G. है ना? यदि ये केस है और cut student, suppose it is from here, from the center or from in between there. या फिर किसी एक में ले लो, ये नॉर्मल है और यहाँ पर एक कट है, तो क्या हो गया? Students, if in any one of the gene also it is from across the gene or from the gene locus, then it is possible that some segment of that gene it will be separated. यदि मैं यहाँ से कट कर रही हूँ, so it is possible that, for example, this is a complete locus. Yes, and if it is cut is from here, यहाँ से यदि कट है, so इतना जो पार्ट है ये रिवर्स होगा और ये वाला जो पार्ट है it will remain as it is. Yeah, so some part of that gene will be separated and the other will remain there as it is. So because that will be separated, because of that now it can show some abnormality. Yes, because here cut is in between the gene. It is not from there where the gene is not involved, but here it is in between the gene. So some segment of that gene is going to be lost. Yeah, suppose there is some protein there and then protein is lost, so that protein is not going to be formed. यदि protein form नहीं हो रहा है, so definitely there would be some abnormality. Okay, so such inversion, it can cause abnormality. Such inversion will give you the normal response, and that's why I told you this point, that it is not necessary that all inversion causes damage. Now moving for the next part. Next we can also see that the inversion, as I told you, they are the paracentric and as well as pericentric. इसी तरह से inversion can be homologous, yes, or homozygous, sorry, as well as your heterozygous also. Okay, so homologous chromosomes, if there are there with identical inversion, दोनों chromosome में inversion हो रहा है, these are the homologous. Yes, same है दोनों. Yes, they both are same. If inversion is here, here also inversion is here. So with identical inversion in each member, there will be no problem. It will undergo normal distribution, है ना. जैसे यदि ये homologous है और ये भी homologous है, and inversion is in this part, and here also inversion is in this part. So यदि ये वाला 3 4 5 जो है, ये हो गया आपका 5 4 3, तो ये भी हो जाएगा 5 4 3. So there is no problem. 5 is with 5, 4 is with 4, and 3 is with 3. Just their place has been changed. So they will undergo a normal distribution. However, if the crossing over in an inversion heterozygote, जैसे कि मैंने बताया कि एक आपका नॉर्मल है और उसका हेट्रोजाइगट जो दूसरा रहेगा होमोलोगस, वो क्या रहेगा, it will be defective or inverted, then it can produce deletion, duplication and other curious configuration. Okay, so on that basis you can say that the inversion can be homozygous as well as it can be heterozygous. Yes, now what is a homozygous? If the inversion happens at same position, inversion at same position, on both homologous pair, then what happens? Then during recombination, जब recombination होगा after crossing over, normal response is seen. Yes, like if I am talking about, like suppose this is a homologous chromosome, है ना. Now if this is A B C D E C D E, if the inversion is here, suppose in D and E, so I will make it as E and D. If suppose this is inverted और यहाँ पर भी ये भी inverted है, yes, this is also inverted, that means what? There is no problem because it is same. So in this case there is no problem. Got it? So this is homozygous inversion. If it is a heterozygous inversion, that means if the inversion in only one pair and not in another, inversion in one pair and other is normal, then such is called as a heterozygous. Yes, then during recombination में क्या हो जाएगा? During recombination there will be problem can be arise. Yes, there could be some sort of abnormalities. Yes, क्योंकि क्रॉसिंग कैसे हो जाएगी? It will be unequal crossing or unequal crossing. How it would be unequal crossing over? For example, again you can see here, suppose the same thing A B C D E C D E. ठीक है, यहाँ पर A B as it is है, C the inversion is here in E and D. This is inverted, but here this is normal. So here this is inverted and this is normal. So what happens during crossing over? There will be some problem. Yeah, so it will cause unequal crossing. Yes, it will show you the unequal crossing क्योंकि ये दोनों अलग अलग है ना. So this is what is called as a heterozygous and the homozygous inversion also. I hope this is also very much clear. Yeah. Now coming to the previous one जिसमें हमने बोला था the paracentric as well as the pericentric. Yeah. So paracentric में I think I will explain you from this figure only. This is nothing but, now tell me whether it is an paracentric or pericentric from the figure only. You can see here, this is a normal chromosome. Yeah, this is a normal chromosome A, B, C, D, E, F, G, H. And here you can see A, D, C, B, E, F, G, H. So there is an inversion in this case. Here, ये वाले पार्ट में इन्वर्शन है. और सेंट्रोमियर कहाँ पर है? सेंट्रोमियर ये है और ये भी ये है. तो जो इन्वर्शन हो रहा है उसमें सेंट्रोमियर इन्वॉल्व नहीं है. Centromere is not involved. So which type of inversion it is? First of all, this is what we are looking is a paracentric inversion. है ना? This is paracentric inversion. Now listen and carefully students. If I am talking about the paracentric inversion, यहाँ पर this is inverted chromosome, this is a normal chromosome. Now, when this go for a prophase 1 in the meiosis students, yes, in meiosis to prophase 1 if you will see, what happens, they will go for a crossover. उसके लिए जब जाएंगे तो we know that there are 2 for this and 2 for this, है ना? ये दो पेयर रहेगा, ये एक पेयर रहेगा. तो ये नॉर्मल के 2 हो गए और ये इन्वर्टेड क्रोमोसोम का 2 हो गया. Isn't it? Now, no need to tell you why this 2 and 2 are there. We have all done this during the division and cell division, and there you all know. And that's why I am saying it is during the prophase 1. So, the crossing over it will take between 2, yes, which are in the center. ये वाला एक बार वाला और ये बार वाला तो इन्वॉल्व नहीं होगा क्रॉसिंग ओवर. या तो जब ऐसा क्रॉसिंग ओवर हो रहा है कि एक नॉर्मल है और एक में इन्वर्शन है, सो क्या हो जाएगा स्टूडेंट्स? इफ आई वांट टू मेक दिस बी कनेक्टेड विद दिस बी, या इन नॉर्मल केस इफ दिस इज स्ट्रेट, दिस बी विल बी हियर. या so that's why the loop is going to be formed so that this could be proper crossing can be done. ठीक है, so crossover हो रहा है. During the crossover, loop is formed. Yes, after the formation of a loop, there will be a crossover here. You can see here it is a crossover. तो अब देखो crossover का result क्या है. Now listen carefully. The first part that I am telling you, this is the normal one. Because this is not taking part in a crossover. Yes, so this will come as it is normal. You can see A, B, C, D, E, F, G, H. Yeah, so this is a normal. Now the second one, it is going to take part in a crossover. So you can see A, B, then it is going for a crossover. This will go here, यहाँ पर जाएगा C, yes. Then in this way it will move D and then A. देखो ये देखो A, B, C, D and A. बराबर है? So what can you see in this case students? यहाँ पर yes आप देख सकते हो. Now after that the third one, थर्ड वन कहाँ पर है जिसके बीच में क्रॉस ओवर हो रहा है, ये हो गया, ये आ गया. Now if I am talking about this, so it will go like how? It will be A. किस किस के बीच में क्रॉसिंग हो रहा है, ये हुआ तो I will take this also again. Yes, I'll take this one. So what can I find in this case? The fourth one, if I am taking it, again we can take it as A. और यहाँ से देख लो आप H G F E then B. Yes, after crossover where it is moving? Can anyone tell about that? If you come on, I'll again explain you. I think this much is clear. ये पहला वाला जो हमने देखा, this is normal. This is normal that we have seen. This is the second one that we have seen. Isn't it? How the third is formed? How the third is formed? And in both the cases, can you see? यहाँ पर this is one centromere which is formed. In this case, here is also a centromere and here is also a centromere. Two centromere आ रहा है. Isn't it? Isn't it? So this is the crossing over between 2 and segment 4, because this is a 4 segment whose loop is there. If I talk about now 2 and segment 3, so what can you see in this case? यहाँ से हुआ, now you will see the crossover from here. I am making it with a different color. HGF, yes, HGFE, D students. Then it go to C. Again there is a crossover, so it will come down. Then B, E, F, G, H. देखो ये रहा H, G, F, E, D, C, B, E, F, G, H. ये वाले जब after this whatever it is forming, can you see any centromere here? In this crossover there is no centromere involved. Centromere तो आया ही नहीं है. यहाँ पे 2 आ गए, यहाँ पर एक भी नहीं आया. Yes, and the last one you can see here जो कि ये था. Yes, this is as it is A, then it is D, C, B, E, F, G, H. A, D, C, B, E, F, G, H. Can you see this? This will be the result after the crossover. Yeah, so what can you see? One is becoming acentric and one you can get dicentric. अब ये जब यहाँ पर break होगा, when there will be the break after the crossing over, separation होगा. So this is separated, so you will get one normal product. Yes. Now there will be a segregation at NFS1 and segregation at meiotic stage. So when there is a segregation at NFS1, what will happen due to the stretch? इन दोनों के बीच में पहले ये दोनों ब्रेक होगा, तो यहाँ पर stretch आएगा, and at any point there will be a break in this segment. For example, if the break is from here, yeah, और ये तो acentric है, उसमें centromere ही नहीं है, so it will be lost there only. Yes, this fragment it will be lost. Got it? तो ये तो लॉस हो जाएगा, तो ये वाला जो रहेगा, ये देखो क्या होगा, this will be the first part and normal. Due to the break till here there will be D, यहाँ D तक रहेगा, और ये वाला part जो सिर्फ A वाला, it will remain with this, and this is the last one. This acentric it will be lost because there is no centromere. Centromere नहीं रहेगा, तो how the segregation it will be there, isn't it? So what we get after the result that we get, one is normal product, second is the deletion products. This is also deleted because A, B, C, D hai. E, F, G, H is not there, so it is deleted. Here also this is deleted, only A is there. So these both are the deleted product, and this is normal, students, but what can you see? D, C, B is there. That means it is an inversion product. genes तो सभी present है, but they are inverted. So this one is a normal product there, and you can get one is your inverted product. Yes, and two are defective. Two are defective due to the deletion. This is a result for a paracentric inversion. समझ में आया? So in a paracentric inversion, what we have seen in a paracentric inversion, we have seen that there will be one dicentric. ये आपको पूछते हैं एग्जाम में. Yes, one acentric fragment is going to form. This acentric fragment is lost. Yes, and this dicentric product forms two defective. Yes, in case of an inversion. So what is the result? Our result is of the paracentric inversion का result क्या आएगा? One normal, one inverted and two defective. So remember, this is the result. And why this is paracentric? आपको ये भी समझना पड़ेगा फिगर देके कि paracentric है कि pericentric है. Yes, so this was paracentric. Now moving for the next. Now you can see in this figure आप क्या देख रहे हो? सबसे पहले ऊपर देखो कि क्या है A B C. Yes, then here it is. This is the centromere. D E F G H, and here A D C B. So the inversion is there, students, and inversion is including the centromere. So this is what we call it as a pericentric inversion. क्या है ये? It is pericentric inversion. Okay, come on. We will discuss now what happens in this pericentric inversion once again. So again, this is a normal, this is an inverted or inversion. Now during the prophase 1 of your meiosis, they will undergo for a crossover. ये जो homologous chromosome है, am I right? Because there is an inversion in the second chromosome, तो ये second, third और fourth जो है, they will undergo a formation of a loop. Yes, loop formation होगा. Same thing will happen. ये पहला वाला आपका जो है which is not taking part, it will, what it will do? It will become a normal. Yes, it will become a normal. Now the second one which will take part here, what you will see, A, B, C. कहाँ पर हो रहा है crossover? Crossover is happening here. Just a minute, I will make mark there. Yeah, crossover is here. Yeah, so what I will say now, A, B, now it will move down, नीचे जाएगा, C, then it is your what, D and A. Can you see there? बराबर है? तो इसका centromere कहाँ था? Is the centromere involved here? For this, the centromere is not involved, but yes, in this case, ये second वाले का, that means इसका centromere यहाँ पे रहेगा. So your second segment, it will be what? A, B, C, D, A. Isn't it? Now if you will see the third one, what can you see in this case? आप देखो, A, कहाँ पर crossover हो रहा है? If I talk, anyone can tell me कहाँ से crossover होगा? Yes, if I take 2 and 4, so you can see here H, G, F, E. Yes, then it will go here. In this way, this will come. It will come here and it will move like this. तो क्या आ रहा है आपका? H, G, F, E, B, H, G, F, E, B, C, D, E, F, G, H. Yes, C, D, E, F, G, H. Got it? This is a centromere in this case. यहाँ पे भी centromere है, इसमें भी centromere है, इसमें भी centromere है. और ये जो बच गया, it is A, D, C, B, E, F, G. This will be third one. This one. Because this will come as it is. So all of them, they are having the centromere. So that's why in this case there is no acentric or dicentric fragment. In pericentric inversion there will be no acentric and dicentric fragment. Remember this thing. Remember, there will be no acentric and no dicentric fragment. Why? If I am asking you the reason why, because students, a centromere is involved in this case. Centromere present in each strand there. Isn't it? Centromere is present in each strand. That's why there will be no acentric or dicentric fragment. Each one centromere is present there. Okay. Again the result, what can you see? यहाँ पे result क्या मिल रहा है आपको? एक आपका normal है. Yes. दूसरा भी आपका normal है लेकिन inverted है. ये वाला. This one. Yes, this is normal. This is normal but inverted. And other two are again not normal, I can say defective है. That is inviable, this and this. Yes, as a deletion or the duplication. In this case, there is a deletion. इसमें deletion है. और यहाँ पर क्या है? यहाँ पर duplication है. Yes, और deletion भी है. Deletion क्या है? We can say deletion and duplication. Here also duplication is also seen. देखो यहाँ पर A duplicated है. ये वाला A है. I think you can see here, this is A and this is A which is duplicated, and other product, that means EFG, it is not there. So deletion and duplication. In this case, A is not there, but yes, HGFE, HGFE, these are duplicated and A is missing. So there is a deletion of A. So again, one normal, one inverted and two defective. But remember, there will be no acentric, no dicentric after the crossing over. Is this much clear? तो ये था आपका inversion जिसमें आपका now remember, on this paracentric and pericentric inversion many questions are formed. फिगर के बेसिस पे भी क्वेश्चंस पूछे जाते हैं, स्टेटमेंट बेस पे भी क्वेश्चंस पूछे जाते हैं. तो योर कंसेप्ट शुड बी वेरी मच क्लियर. Got it? Definitely once we do this chromosome mutation part now, I'll also take some classes for questions, general questions which are asked on this on this topic. So आपका practice भी हो जाएगा, isn't it? Come on. Now we will move for the next, and next is translocation. Okay. Now what do you mean by translocation? Translocation क्या है? Okay. Before translocation, I will also add one more thing. I have not given you any examples for the inversion. है ना? Inversion के the examples लेना है. So what would be the examples of inversion? A very common, if you know, students, chromosome 12. Yes, inversion of chromosome 12 क्या होता है पता है? Yes, this has created the tallest teenager in the world. That this tallest teenager which is there, it is having a inversion in its chromosome 12. Got it? So this is a very good example. One more example you can take of the, there is a species, insect species is there, Frigida, yes. It has a variation of chromosomal inversion that allows the species to make series of physical differences. Yes, so I can write here the species what it has a variation of chromosomal inversion that allow the species to make series of physical differences. तो ये म्यूटेशन से क्या होता है? दिस म्यूटेशंस gives the insect variety of physical attributes, है ना? यानी बहुत सारे फिजिकली चेंजेस आपको दिखेंगे वो एक ही स्पीशीज में. दिस इज ड्यू टू द mutation or due to the inversion there in this species of insects. I hope this is clear. So now moving back again for our next part, that is translocation. Translocate करना, एक जगह से दूसरी जगह जाना. Translocate, that is nothing but your translocation. That is the shifting or transfer of a part of chromosome or a set of genes to a non-homologous one. Now again there can be, यहाँ पे भी दो प्रकार से हो सकता है. एक ही क्रोमोसोम में एक जगह से दूसरे जगह पे शिफ्ट करो, या फिर एक क्रोमोसोम से दूसरे क्रोमोसोम पे जाओ. Both are the translocation. Yeah. So again in a simple way we can make it as a shifting or transfer of a chromosome or a set of genes. Now where it is shifted, accordingly we can give the name of to the translocation. Now in translocation also there is no deletion students, कोई भी loss नहीं है. There is no addition or duplication नहीं है. Yeah, we can say there is no rearrangement also, just the change in the place. Yes, there is a change in the place. That is a rearrangement I can say, but rearrangement also of the position from here to here. Yeah. So the genetic significance of the translocation is that the genes from one chromosome are transferred to the another. Clear? Now, translocation can be again, as I told you, तीन तरह के हो सकते हैं डिपेंडिंग अपॉन कि जो जीन ट्रांसलोकेट हो रहा है, ये सेट ऑफ जीन जो ट्रांसलोकेट हो रहे हैं वो कहाँ हो रहा है. So accordingly we can say that it could be reciprocal. Yes, reciprocal translocation, non-reciprocal translocation, और तीसरा आप बोल सकते हो shift translocation. So what are this reciprocal, non-reciprocal? Now ये भी जो हो रहा है आपका, it can be again, we can say that it could be on the basis of interchromosomal or intrachromosomal. Yes, the rearrangement can be interchromosomal or intrachromosomal. As I told you, एक ही क्रोमोसोम में भी हो सकता है और अलग अलग क्रोमोसोम में भी हो सकता है. तो on this basis you can say interchromosomal or intrachromosomal. इसी के बेसिस पे हम ये तीन प्रकार देख सकते हैं. That is reciprocal translocation. So now what is this reciprocal translocation? Yes, so when a piece of two non-homologous chromosomes are interchanged without any net loss of genetic material, we call it as a reciprocal translocation. Again I am repeating, piece of non-homologous chromosomes, दोनों chromosomes कैसे हैं? They are non-homologous, they are not same students. Yeah, like for example if I am saying just this is A, B, C, D, E. Okay. And here I can write P, Q, R, S, T. So this is a non-homologous chromosome. Yeah. Now if there is a reciprocal translocation between these two, how I can say, or what is the reciprocal translocation? Suppose this part, that is this B, is translocated. Yes, and the same thing what happens, what will be the next thing here? From here suppose this S is translocated. So the new pattern what I'll get? I'll get A, S, C, D and E, and here I will get P, Q, R, B and T. So this is called as reciprocal. ये यहाँ का यहाँ गया और यहाँ का यहाँ आया, isn't it? So this is what is called as a reciprocal translocation. अब ये दोनों नॉन होमोलॉगस क्रोमोसोम है ना, यानी एक क्रोमोसोम से दूसरे क्रोमोसोम में गया. So in this case, a segment from one chromosome is exchanged with a segment from another non-homologous one. So that in reality, two translocation chromosomes are simultaneously achieved. Those translocated chromosome मिल गए ना, यहाँ पे भी translocation हुआ, यहाँ पे भी translocation हुआ. Whereas in non-reciprocal chromosome or non-reciprocal translocation, what happens, students? The transfer of genetic material from one chromosome to another without any reciprocal exchange. That means यदि मैं बताती हूँ, suppose this is A, B, C, D, E and again this is P, Q, R, S, T. Yes, A, B, C, D, E and P, Q, R, S, T. Okay, in this case, if there is a translocation and if I say this AB is translocated, ये AB translocate हो रहा है, तो क्या हो जाएगा यहाँ पर? This will translocate here, so the result it would be यहाँ पर सिर्फ C, D, E बचेगा और यहाँ पर जो है it would be your P, Q, then R, S, T, A, B, all will be there. Are you getting it? Yeah. So this is what we can say. You will get this. ये हो जाएगा आपका non-reciprocal. Yeah. One more example I can tell you about the non-reciprocal on a simple way. Suppose consider two non-homologous chromosomes. Yes. इस तरह से यदि नहीं समझ रहा है तो I can also make you like suppose this is two non-homologous chromosome A, B, C, D, E, F, G and the second is M, N, O, P, Q, R, S. This dot is nothing but the centromere. Now, if the chromosome segment EF, this EF I want to move from first chromosome to second chromosome without any transfer from second to the first. So that would be non-reciprocal chromosome. यदि इसका result आता है A, B, C, D, G and M, N, O, P, E, F, yes, then Q, R, S. So what is this? This is your non-reciprocal. समझ में आया? यानी एक जगह से शिफ्ट हुआ बट दूसरे से वापस उसको नहीं मिला क्या? And if I same thing, if I am talking about this, इसी की यदि बात करें हम, and if the translocation is like that, A, B, yes, C, D, and if the translocation is here, so this will also be there. So यहाँ पर suppose Q, R, G, Q, R आ गया, C, D, Q, R and G, and in this case you can see M, N, O, P, E, F, S. So this both are, I can say, you can see here दोनों एक दूसरे को, इसने इसको दिया, इसको इसने दिया. So this is what is called as a reciprocal translocation. I hope this is also very much clear what is reciprocal translocation and what is non-reciprocal translocation. One more is there shift translocation बोलते हैं. यहाँ पर क्या होता है? It is not the different one, but the broken segment of one chromosome gets inserted interstitially in a non-homologous chromosome. समझ में आ रहा है? Like suppose if I talk about the shift chromosome, how can I, where I can explain? मैं यहाँ पर आपको explain करती हूँ with a different color. I think so. Just a minute. Yeah, we can say like this, students. If I am talking about the shift one, I don't have that much space here, but yeah, suppose this is your A, B, C, या फिर हम उसको without drawing only I will write it down. If suppose it is A, same thing if I am taking A, B, C, D, E, F, G and M, N, O, P, Q, R, S. Okay. Now if students in this case this B is translocated, तो क्या हो जाएगा B का ट्रांसलोकेशन, ये इंटरस्टिशियली बीच में घुस जाएगा. लाइक यहाँ पर तो आपका हो जाएगा or you can not take, just a minute, I will make you one more thing here. Yeah, so it will move here interstitially, so it will become what? A, yes, dot, then what will be there? यहाँ पर आपका आ जाएगा C, D, B, E, F, G. So this is what is called as the shift translocation. ये क्या हो रहा है? It is in a non-homologous chromosome. One chromosome get inserted interstitially in a non-homologous chromosome. Got it? इसी को, because there was no space, so I have rubbed the second part. You can also make MNOPQRS and that B will be inserted there. So it would be MNOPBQRS in that way. But in between it will go there. So that is called as a shift translocation. Got it, students? So ये था आपका translocation. You can see here the figure. This is what is called as a non-reciprocal intrachromosomal translocation. Intrachromosomal, that means ये same AB, ये BC यहाँ से translate हो रहा, वहीं के वहीं आ गया. Am I right? This is interchromosomal, again non-reciprocal. This is BC, it is translocated here. From here it is deleted completely. And this is your reciprocal interchromosomal translocation. This is reciprocal. This is AB is shifted here and MNO is given here. Isn't it? देखो ये हो गया आपका reciprocal. It is very simple. I hope this is clear, students. Yeah? Chalo. So now the next we will move there. This intrachromosomal, you can also call it as a shift translocation actually. Now we will move for one more translocation, that is called as Robertsonian translocation. अब क्या है? What is Robertsonian translocation? Students, the non-homologous chromosomes, when they fuse at their centromeres. Repeating, non-homologous chromosomes, when they fuse at their centromeres, they create a structure called as a Robertsonian translocation. This name was given for the cytologist F.W. Robertson, who had given this concept. Okay. For example, I can say if two acrocentric chromosomes fuse. Acrocentric, what is their acrocentric? I think you all know what is acrocentric. One arm long, one arm short, and where is the centromere? Centromere is present near the tip, isn't it? So that is the acrocentric. So the long arm of two acrocentric chromosomes becomes joined to a common centromere. ठीक है? Through translocation, generating a metacentric chromosome with two long arms, and another chromosome will become with two short arms. Yes, this is what is called as a Robertsonian translocation. The smaller chromosome often fails to segregate, leading to an overall reduction in the chromosome number. Yes? That means Robertsonian translocation क्या हो रहा है? It involves generally the chromosomes which are acrocentric. तो कौन कौन से acrocentric chromosome है? Remember, chromosome 13, 14, 15, 21, 22. These all are the acrocentric chromosomes. So they can all show the Robertsonian translocation. Yes. Why? Because their ends, yes, the ends of the short arms, they have similar repetitive DNA sequence. Yeah, that's why these all are, or they can show the Robertsonian translocation. समझ में आया कि नहीं समझ में आया? For example, if I say here, if suppose this is the acrocentric chromosome, as I told you, है ना, one arm short रहेगा और one another long रहेगा. Suppose I am taking this as a chromosome 13 और दूसरा हम लेते हैं chromosome 21. Suppose they both are the acrocentric. Yes. If here the translocation is done, तो यहाँ से कट होगा. Yes. So this long will fuse with long and long will fuse with long, short will short, short with short. So what you will get? You will get this way. आपको रिजल्ट ये मिलेगा. समझ में आ रहा है? Two with both short arms and two with both long arms. Yeah. So one is with very long and one is short. तो ये generally what होगा? They fail to segregate. Yes, when they will fail to segregate the chromosome, there will be reduction in the chromosome number, isn't it? So this is what is called as Robertsonian translocation. You can see here. This is two acrocentric chromosome, students. There is a crossover. यहाँ पे crossover हुआ यहाँ पे. You can see here. ये और ये fuse हो गए, ये part और ये part. ये यहाँ पे देख सकते हो. और ये वाला part और ये वाला part साथ में आ गया. तो this is, तो ये lost हो जाएगा because it is very small. और ये बन गया आपका metacentric Robertsonian translocation. Got it? Metacentric, centromere center में आ गया. Both the arms are long arms there. I hope this is clear. So कौन सा Robertsonian translocation कौन सा ऐसा chromosome दे सकता है? The acrocentric chromosome. I had given the numbers also. It is 13, 14, 15, 21 and 22. Clear? Now some examples of translocation. कौन से examples हो सकते हैं translocations के? Can anyone give me the answer for that? Which are the different examples? Can you think about that? A very good example is, first of all, you can say this can be due to the chromosomal non-disjunction, non-disjunction, translocation of chromosome 21. Chromosome 21 onto the chromosome 14. 21 translocate कहाँ करेगा? 14 chromosome में, and this causes Down syndrome. Yes, ये इस तरह का translocate यदि होता है 21 chromosome का 14 chromosome में, yes, due to the non-disjunction, students, it can lead to again a syndrome called as a Down syndrome. Okay. One more example of translocation, you can make it as a chronic myeloid leukemia. इसमें क्या होगा? This is done by the translocation of genetic material between chromosome 9 and chromosome 22, students. Yes. These such chromosomes are called as क्या? Philadelphia chromosomes. Yes, this Philadelphia chromosome, they forms when part of chromosome 9 attaches to chromosome 22. ये chromosome को हम क्या बोलते हैं? We call it as a Philadelphia chromosome. Isn't it? One more example you can also make. Philadelphia chromosome बोलने से और ये बनने से क्या होता है? The disorder that it is causing, students, this disorder causes formation of tyrosine kinase. Yes, and this tyrosine kinase definitely helps. It is a kinase there, isn't it? So it helps in the cancer cell growth. That's why it is chronic myeloid leukemia. Got it? Now there is also translocation between chromosome 8 and 11. और ये ट्रांसलोकेशन कॉज करता है स्टूडेंट्स एक्यूट माइलॉइड ल्यूकेमिया. These are some examples of translocation. You can find many others also. मैं आपको कुछ important examples ही देती हूँ. Yes, there are so many. You can find it all that. I hope now this concept of inversion and translocation is very much clear to all of you. Yeah, in the next class we will move for the next type of mutation again. It will be the chromosomal but on the basis of numbers. Yes, so what is euploidy? What is aneuploidy? ये सारी चीजें हम लोग next class में पढ़ेंगे. So we will stop here, students. I hope you like the class. If you like the class, you can like the video as well as you can subscribe the channel or the Academy official, and also you can share it. Yes. Also we have an application, that is Arth Academy app, students. You can download this app from the Play Store. The link is also given in the description box, because in this app you will find some courses which could be useful for you along with all the video lectures as well as the notes and the practice questions for the particular courses there. Yes, so I hope you enjoyed the class. Thank you, students. Take care. See you soon in the next class. Keep learning. All the best for your studies. Yes, this is the link of the Arth Academy app. Also, you will have that link in the description box. So please like, share and subscribe the channel. Till then, all of you, bye bye. Take care.